All right, are there any questions? Sorry that the lecture ran about 2 minutes over.
I got questions, but about the unknown project. I'm completely lost.
Okay. If you're completely lost, we'll probably have to talk either after the lab or during the lab, because I don't think I'll be able to explain to you enough here to get you totally unlost. Sorry, I'm seeing a whole bunch of ants in my place and I'm killing them. But if you have something quick, I will certainly address it.
Yeah, let's talk after lab. Thank you.
Okay. All right. Any other questions? If not, let's go ahead and get started. Let's see, I'm recording. Let's go ahead and share the screen. So if we look on the schedule, oh, I need that. We're covering Lab 7 today, the throat and urine cultures. And you'll also start your work on your unknown as soon as I send you the information. So I haven't sent you the results yet. I'll try and do that after the lab. Hopefully I'll get that done tonight. Any questions? Once you get your results, look over your results and look at the table of the species. I don't remember what it's called, but there's a link to it. There's a chart of the unknowns. And then consider what possible species you have, and then look at what test you want to do. If you run more than one test, you're not using at one time, you're not using dichotomous logic, and you will be penalized. So you want to use dichotomous logic and ask for one test at a time. All right. I don't really have anything more to say about your unknown work other than that in your unknown notes, that's where you're putting your notes for the unknown project. You should have up there your unknown number, and you should put up there that you've started the that you did the fermentation test yesterday. You did the gram stain on Sunday, and you did the gelatin hydrolysis test last Tuesday. And you should get the results tonight after I send them out. And once you get the results, you should put in what your results are for each of those 3 tests. Okay? So that's all you should have in your unknown notes page, at least at the moment.
So are we supposed to be submitting that to you like on a daily basis type of thing?
You should be adding that in your unknown notes. You're not submitting it to me, but you do submit it as a document whenever you add something. Don't put it in as a comment. Add it as a document, and then I will look at your documents that you've submitted.
And is that the only way that you're going to be sending out? Because I have what you said is completed. I just haven't been submitting it in the thing thus far.
So you have it on your own computer, you're saying,
yes,
but I can't see it.
Correct, yeah.
Okay, well, that won't do you any good. I can't grade it if I can't see it. So it needs to be submitted in your unknown notes. Most people won't do it every day. They will be doing it maybe a couple times a week at most.
And that's fine. I'll submit it to you is what I'm saying. But if I submit it like tonight, will I still get those results tomorrow, like you said?
What results tomorrow?
The results for the the tests that I have in my unknown notes for the.
Yeah, you can write in your notes that you've done it, and then I'll submit the results to those 3 tests. Not tomorrow. I will submit it tonight, and then you can add in what your results are. That could be even tonight, or you could do that tomorrow. I don't care. That's up to you when you submit it.
Okay, that's fine. I just want to make sure I wasn't going
Okay.
to miss out on those 3 tests.
Yeah, you're not going to miss out on those 3 tests, and you won't miss out on any points. I've got the date when it's graded. It's near the end of the term. As long as you have everything up by that date when it's due, you won't miss any points. My point is that if you ask for a test and you do not have your unknown number submitted, you're going to get a note saying, I can't submit it. I don't have an unknown number for you. Okay? So you do
And
need to record your stuff in the unknown notes.
what were the 3 tests? You said the gelatin test, the gram test, and what was the third one?
It should be the
Lactose fermentation.
Fermentation, yeah.
All right. That was a few more questions than I was hoping, but that's all right. I don't think I need this window open, so I'm going to shut it. And we'll leave that one there. And I've talked about the what we're covering and the unknown notes. So let's move on to Lab 7, the throat and urine cultures. There is some reading that should be done before this lab. Not very much, but a little bit. There's some supplemental videos and a virtual lab. And I'm sorry, I have not found anything other than Flash Virtual Lab. So just do your best to run it. And if you can't run it, well, you can't run it. There's nothing else to do. Primary learning objections. You should be able to explain how to properly collect throat and urine specimens for routine culture. You should be able to list the common bacterial pathogens isolated from the throat and urine cultures. You should know the culture media used to cultivate these pathogens coming from the throat and the urine cultures. You should be able to recognize which tests are used to identify a throat and urine pathogen. And you should know the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will talk about the XV factor testing, but I'm not going to test you on that. Once specimens are received in the laboratory, they are logged and processed in timely fashion. Specific culture media are inoculated and incubated, and specific culture media for throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from these cultures. For the throat, we frequently grow things on blood agar, but sometimes on chocolate agar. Streptococcus pyogenes is a common bacterial pathogen, and it grows on blood agar. Haemophilus influenzae, which is seen most frequently in children, especially young children, which we use chocolate agar on. For urine cultures, we use blood agar, and either McConkey's agar or EMB agar. EMB is eosine methylene blue, and usually it's either McConkey's or EMB, not both of them. Some.
Are you eating, Junior? Put that butterfly down. Actually, give it to me.
Let me mute that. That wasn't me, by the way. So, for urine culture, some common pathogens include various Streptococcus species, Enterococcus species, and Staphylococcus species. The one main problem, or one main species of Staphylococcus that causes disease will be Staphylococcus aureus, and we'll talk a little bit about that. And then there's also Enterobacteriaceae, like E. coli, which is a urine pathogen, and then Pseudomonas. And there's many species of the Enterobacteriaceae and a few of the Pseudomonas that can be a urine pathogen. All right, so for throat cultures, sore throats, you can look at it and then usually determine whether it's viral or bacterial in origin. If there are white patches on the inflamed tonsil, it can be indicative of a bacterial infection. And you look at the back of the throat or the tonsils, and if there's white patches, it might be bacterial. If it's only red and swollen, that's likely a viral infection. The most common bacterial pathogen in the throat is Streptococcus pyogenes. It does have another name, which I do not use, Group A strep or Streptococcus. It's funny that the United States uses the name I don't use, but I use the species name. Strep throat is done by swabbing the back of the throat or the tonsils where there's redness or white patches. You do not want to touch the tongue or the cheek or the top of the mouth. You only want to go way back in the throat to get a throat swab for testing for a throat pathogen. So unsuitable, the sides of the mouth and the tongue. The tonsils and the very back of the throat is where you want to swab. Patients who test positive for Streptococcus pyogenes are placed on antibiotic therapy. The reason is that Streptococcus pyogenes generally cannot be defeated by the host immune system. And so you need antibiotics to throw off a Streptococcus pyogenes infection. The reason is that Streptococcus pyogenes creates a capsule, and the host immune system really has a hard time fighting Streptococcus pyogenes because of that capsule. Streptococcus pyogenes is fastidious, so it will not grow on general purpose media. You need a media supplemented to grow an enrichment media. You need something like blood agar plate or BAP, and that's supplemented with 5% sheep blood, and the blood cells themselves give the media its red color and the nutrients needed by Streptococcus pyogenes. And then the plate is examined for small, clear colonies surrounded by a complete zone of hemolysis, which is shown here in a minute. And that's indicative of beta hemolytic colonies, which could be Streptococcus pyogenes. And let's see if I can blow this up. All right, so on this plate, do you see that whitish center? It's a real white, very tiny colony, and it's surrounded by complete beta hemolysis, which is white, and the media itself is red here. And so from the media we come up to here, and then it's got a dark ring, that's alpha hemolysis, and then it's a white ring, that's beta hemolysis, and then in the very center right there, that's the Streptococcus pyogenes, or the beta hemolytic colony. Any question about that? A normal throat microbiota which has no beta hemolysis, over here there may be partial hemolysis. And you can see partial hemolysis in a normal throat. Went a little too far. Let's do it there. Only 2 biochemical tests are required to give a definitive identification that is Streptococcus pyogenes when we have beta hemolysis on a blood agar plate or a BAP plate. The 2 tests are doing the catalase test and the bacitracin susceptibility test. So after getting this beta hemolysis on the blood agar test, you then run the catalase test. Catalase, you use hydrogen peroxide, and in the presence of catalase, it breaks down 2 hydrogen peroxide molecules into 2 water molecules and 1 oxygen molecule. And that oxygen in the water and the hydrogen peroxide creates bubbles. And when you see bubbles, you know that catalase is present. And there is a catalase positive species, and there is a catalase negative species, no bubbles. Do you see that? Streptococcus and Enterococcus. It's not, well, I guess it is. This could be Streptococcus pyogenes. Streptococcus pyogenes is catalase negative. It does not have catalase. We then run the bacitracin test. Bacitracin is an antibiotic, and you streak out your bacteria on a region, and then you place down, so you put the bacteria down, then you put the bacitracin on a disc down, and if you have a zone of inhibition around the antibiotic, then we say it's positive for the group A strep test, or it's sensitive to the bacitracin. If it's negative to the bacitracin, then you don't see the zone of inhibition around the antibiotic disc, or it's very, very slight. It's hard to see if that's a little bit of zone of inhibition there or just no bacteria in that slight region there. Like the disc, when you put it down, you may have slipped it. Streptococcus pyogenes is sensitive to the antibiotic, so we should see a zone of inhibition around the disc. And if you see no sensitivity, then you can assume it's not Streptococcus pyogenes. And those are the 2 tests you need to determine whether it's Streptococcus pyogenes or not from a beta hemolytic bacteria growing on a blood agar plate. Another clinically significant pathogen is Haemophilus influenzae, which is a gram-negative coccobacillus. It is most commonly seen in the throat, the ears, and the eyes, or the sinus infections in babies and young children. Haemophilus influenzae is fastidious, but it will not grow on blood agar because it does not lyse the blood. And so a blood agar plate or a BLAP plate will not grow Haemophilus influenzae. Instead, you can grow Haemophilus influenzae on CAP, a chocolate agar plate, and this is the same as the blood agar plate, except that the blood was added when the agar was hot, and it lysed the blood, making it brown, and lysing the blood cells makes the nutrients available for Haemophilus influenzae to grow. It will not grow on a blood agar plate unless you also have Staphylococcus aureus on that plate, and what you do is you have a single streak here of Staphylococcus aureus, and then you grow the Haemophilus influenzae. You streak it going this way on the plate, and as you can see, in this region of the streak, the Haemophilus influenzae does not grow, but it does grow in this region of the plate. And that's because the Staphylococcus aureus lyses the blood, releasing the nutrients, and then allowing the Staphylococcus aureus to grow in this region of the plate. Any question about that? So to determine Haemophilus influenzae, we grow it on a chocolate agar plate or abbreviated CAP. Any question about that? All right, that's it for the throat cultures. Let's move on to the urine cultures. Urine cultures are used to diagnose urinary tract infections, which can be a urethra infection or a bladder infection. Bacteria can travel up the urethra to the bladder, and females are more disposed to a bladder infection than males because of their shorter urethra. When you're doing a urinary culture, the genital area must be cleaned prior to collection. Usually, if you're just collecting the urine, you catch it as a clean catch midstream, involving only collecting the middle portion of the urine. And that way, the bacteria in the urethra will be less contaminating, meaning if you catch it in the beginning of the urine or at the end of the urine. That's one way to do it, and most patients prefer mid-catch collection, midstream, a clean catch midstream collection. But you could also stick in a catheter to the bladder and get the urine that way. And an even more invasive procedure is the suprapubic aspiration, which essentially you put in a needle through the abdomen into the bladder. You obviously clean the abdomen first, and then directly sample the urine from the bladder. This is the way you get the least chance of contamination. This is less chance of contamination, but remember the catheter is going through the urethra, and the lower urethra always has bacteria in it. And then you're sampling from the bladder. And patients don't like either of these two procedures. And you can also use a clean catch midstream, where you do clean the urethra at the outset, flush with urine to begin with, and then catch the urine midstream. Any question about any of that?
Nope.
All right. So once the urine is submitted to the lab, you plate 0.001 or 1/1000th of a milliliter of urine onto a non-selective media such as blood agar plates or BAP. And there's actually an inoculation loop that plates exactly that amount of urine. And I'll show it to you here. Oh, there it is there, sorry. Went a little too far, let's blow that up. So you get a little bit of urine in that loop, and then you plate it on a blood agar plate, and there you can see the bacteria growing on the blood agar plate. Now a blood agar plate will grow many things, not just the pathogen, and it can grow contaminants from the urethra. So the interpretation is complex for when you're doing a urine sample. Let's shorten that a little bit. And additionally to streaking blood agar, you should also streak a selective media that selects for gram-negative bacteria, such as a McConkey's plate, MAC, or eosine methylene blue, EMB. And that's because the most common pathogens of the urinary tract infection are coliforms such as E. coli, and they're gram-negative. So you want to have a plate that selects for gram-negative bacteria. Well, you can read about that. I'm not going to discuss it. So besides growing the urine culture on BAP or blood agar plates, you should also grow it on a selective media such as McConkey's or MAC. The interpretation for a positive urine culture is multifaceted. There are a couple of factors that have to be considered before you interpret your results. The type and collection. For example, if it's midstream clean catch collection, however that's said, versus supraposic injection to pull out the urine, the interpretation is different. Even on the clean mid-catch stream, we expect contaminating bacteria from the urethra. Okay? So you expect to see bacteria. On the other hand, if you take the suprapubic aspiration, we don't expect contaminating bacteria from the sample taken from the bladder. And so if you get bacteria on that, you know that it's a bacteria growing in the bladder, a pathogen. You should also consider the gender of the patient. Males and females will have more contaminating bacteria, depending on which sex it is, from the midstream clean collection, however that's called, clean catch midstream collection of the urine. And then the number of colonies per mil of urine will also determine what's important. You always expect some contamination from the urethra in the if you're collecting from the clean mid-catch collection system, and so you have to take that into consideration. For example, with males with at least 50,000 colonies per mil, and then with females 100,000 colonies per mil, is indicative, may be indicative of a urinary tract infection. If it's under that in males, then you probably don't have a urinary tract infection. But in females, it's got to be under 100,000 before we say it's less than, unlikely to be a urinary tract infection. Any question about how you interpret that? All right, so once you get cultures growing on BAP, it could be many species because many species grow on BAP. It could be Staphylococcus, Streptococcus, Enterococcus, assuming it's a pathogen. You then run a few tests to determine what that pathogen might be. If we're suspecting it could be Staphylococcus aureus, you might do the coagulase test. Coagulase is an enzyme that's used to differentiate Staphylococcus aureus from other Staphylococcus species. Coagulase actually is produced by Staphylococcus aureus and contributes to the pathogenicity of Staphylococcus aureus. What this enzyme coagulase does is promote the protein fibrinogen changed into fibrin, creating a clot. And that helps protect the bacteria from phagocytosis, meaning helps the bacteria from being eaten by our immune system cells, like our white blood cells. Fibrinogen and coagulase creates a fibrin clot, and the next picture should be showing a clot. And there's a clot here, coagulase positive bacteria. No clot here, coagulase negative bacteria. And then you can do it on a slide. You see clumping, and in a positive coagulase test and in a negative test. I'll always give you results from like this. I won't give you on the coagulase results like that. There are two tests for the coagulase test, and I just showed you them. There's the test tube test, and then there's the slide test. The most common bacteria obtained from, I should say, bacteria pathogen obtained from a urine culture is E. coli, and that's in the family Enterobacteriaceae. And to help see E. coli, we plate the bacteria on a McConkey's plate, and because this is growing on McConkey's, we know this bacteria is gram-negative, and it's also fermenting the sugar lactose. So this bacteria is likely in the family Enterobacteriaceae. You'd have to do a few more tests, but that's showing it's there, that it could be the family Enterobacteriaceae. Another bacteria that can cause urine infections or urinary tract infections is the bacteria of Pseudomonas, that family, and particularly the species Pseudomonas aeruginosa can cause a urinary tract infection. It's another gram-negative bacillus, but unlike the family Enterobacteriaceae, which are gram-negative bacilli, Pseudomonas are aerobes, and they do not ferment glucose. So this bacteria here cannot be Pseudomonas, because it's fermenting a mannitol, and Pseudomonas does not ferment mannitol. That's not discussed here, I'm just telling you. What you can do to show that you have a bacteria in the family Pseudomonaceae, meaning a Pseudomonas species like Pseudomonas aeruginosa, is look for cytochrome oxidase C. This is a protein in the electron transport chain that we just talked about in the lecture, and I'm not going to ask you to memorize that, but it's right here and here. And Pseudomonas aeruginosa performs aerobic respiration and has cytochrome oxidase C. And when you do the oxidase test, it's looking for the cytochrome oxidase C, and so we say it's oxidase positive. And the oxidase test, when you look at it, in this region of the slide it's negative, but in this region it turns purple. That's oxidase positive. And we call it the oxidase test, but remember it's actually the cytochrome C oxidase test. Okay? Because we're looking at cytochrome oxidase C. All right? So if it is oxidase positive, we know it can be a pathogen in the Pseudomonaceae family, not the Enterobacteriaceae family, because that family, the Enterobacteriaceae, are all oxidase negative. All right, any questions about any of that? If not, do laboratory exercise laboratory 7. You're not physically going to be performing any of these activities, but you will be responsible for knowing how to perform them. You should know about the CADLACE test, and we have a video and an interactive laboratory. Let's see if I can get that to run. And we have the coagulase test, same video clip, and then the interactive laboratory. The oxidase test. Let's see if that runs. It probably won't run. Well, I might be able to get it if I change the settings. I'm not going to do that later. But you might have trouble with these virtual labs. If you can get them to run, great. If you can't get them to run, well, you can't get them to run. Anyways, there's links for the oxidase test. And then do the questions. Don't do it in the lab manual. Do it in the worksheet. The worksheet is much smaller than the lab manual. And then answer the questions in the worksheet. I will be here until 8 o'clock to answer questions for you. This is the worksheet. And then write in your answer right here. And then submit that to Lab 7 submission folder online in Canvas. All right, any questions? If not, go ahead and get started on the lab. I'll be here to 8 o'clock to answer any questions. And really, you guys, try and ask questions if you have questions, because in the lab answers, some of you have not done a very good job on answering. And I just graded 2 labs, and I'm giving you until next Saturday, midnight on Saturday, to make corrections. As long as you submitted the lab on time, and if you correct your answers and have the right answer, you will get full point on those labs as long as you turn it in by this Saturday. Okay, any questions about that? All right, I'll be here until 8 o'clock. That's 50 minutes to answer any questions. I may go and get dinner, but I'll be back in about 5 minutes. So if I'm not here and answering your questions, just wait until you see me and ask your question then. Remember, to ask a question, you have to unmute yourself. I'll leave mine muted unless I'm cracking nuts, in which case, I'll leave mine unmuted in case unless I'm cracking nuts, in which I'll mute it. Okay. All right, go ahead and get started on the lab. Oh, let's end this.